Good morning, everybody. I'm very glad to see everybody here, and also welcome to everybody that is online. First of all, I just want to thank the DSI for the contribution and that we could actually buy this instrument and that we can have it here at Nelson Mandela University and that we can actually share it with those that want to work with us, and that we can also contribute to good science development. I also want to thank Axion for all their support for us to help us to get the instrument, and also for the team that helped us to set it up the past 2 days. And for everybody that was involved. Thank you very much. I just want to introduce our main speaker that will actually present the presentation and our impedance instrument. So it is Dr. Nicolas Roy that is standing here. Please, I'm so glad that you are here with us. The past 2 days, he really shared his scientific knowledge with us, and it was really a big pleasure to have you with us. So Dr. Nicolas Roy was born and raised in Bordeaux. It is in the south west part of France. His academic background is in virology with a PhD from the Cochin Institute, followed by a postdoctoral research at the Pasteur Institute. During that time, his work focused primarily on HIV and the arboviruses. About 4 years ago, he transitioned into the biotech industry and joined Axion BioSystems. There he specialized in neuroscience and cardiology technology and currently serve as a field applications scientist for the region which includes Europe, Middle East, and Africa. In this role, he has the opportunity to travel widely and supporting scientists across various countries in acceleration and enhancing their research through innovative technology. So I can just say the past 2 days, we have worked closely with the Axion team and with Dr. Roy, and it was really a pleasure, and he can share a lot of information with you. So I will be glad if you put your, those that are present here can ask questions. Otherwise, I'm sure they will be able to put questions in the chat box. Okay, thank you very much. I hand over to you, and then we can discuss the questions later. Thank you.
Thank you very much for a very kind introduction. That's a real pleasure for being in South Africa, second time of my life. I was looking for warm. Today I'm very lucky, so that's great. So thank you. I would like to take a couple of seconds before talking about the instrument. Thank you very much for Professor Wu for your welcome, for all the science we've been running these last couple of days. I think it has been a lot of interaction with you, your team. We did a lot of good things. I will have a chance to make an online last-minute analysis of the data that we gathered together the last couple of days. So I think that will be a very nice way to show you how easy in real time. It will not be fake. It will no preparation. We got the data this morning. I just received them, so we'll make some analysis in real time. That will be nice. So thank you very much for your welcome and all the science and all the moment we shared. Also, thank you very much to our collaborator, Apex Scientific, especially Preston and Krivani, which are here in the audience. So thanks a lot for the organization and your collaboration. Okay. So as you said, so my name is Nicolas Roy. I always say Nicolas Roy. That's actually a French version. It's Nicolas Roy. So I will try to keep my French name. So Nicolas Roy. And I'm a field application manager for Axion. Today, I will talk about the portfolio of Axion, and more particular about the impedance system and the imaging system. So you will see in Axion, we don't have 10,000 systems. We are really focused on a couple of them. The one that we use is impedance system. I will explain you later on what is it about, and also how the imaging can bring a very nice contribution, can bring additional data to this kind of system. Couple of words very briefly on the company. So it has been created in 2008. The company is based in the US, but we have a couple of offices all across the world, one in the US, two in Europe, and one in Asia. We started as a small company, and we made the first EMEA system, EMEA standing for microelectrode array, where we built the first microelectrode system to be able to monitor electrical activity coming from neurons and cardiomyocytes. And then we become one of the leaders in this area. Many employees all across the world, a lot of systems installed, a lot of collaboration. So all these numbers, and a lot of paper, a lot of reviewed paper, all these numbers just to show you that Axion is not just providing system, but is also enabling science all across the world. So I think this system, it does change by itself. These numbers are also very good proof to show how is built the trust between biotech companies and also research institutes. As I said, I will not talk about all the instruments that Axion BioSystems provide. I'm just going to focus on the one that we've been working with the last couple of days, which are the impedance systems. So this impedance system can be described in three different families, what we call the Maestro Z and the Maestro ZHT that you can see on the left and on the right. These two instruments are pretty small. It's like a big PCR machine, I would say, and it's a kind of tiny incubator. So in the room that you can see, you will insert a plate and you can control the environment. You can control the temperature, you can control the CO2. So you don't have to insert this instrument inside an incubator. It's very small, very convenient, and you can run experiments for a very long period of time just because the environment is controlled. The first kind of instrument that we provide is called the Maestro trays. So if one day you need to increase your throughput, if one day one plate is not sufficient anymore, you can jump to the Maestro tray. So this one allows you to run 4 different plates at the same time, but it has to be placed in an incubator. Okay, so 2 bench-side instruments and one that you have to place in an incubator. So there is a lot of specificities for the instrument, which makes it very easy to use. So the main philosophy of a company, regardless what instrument we're talking about, is to allow anyone to make science with them in a very easy manner. So I think people will be maybe able to talk about it, but the only thing that you basically have to do is to culture your plate, insert the plate inside the system, and you just run away. All the calculations, the statistical information will be automatically done by the system. Couple of specificities very quickly. Every plate that you're going to use as a barcode scanner, which makes that every time the system will see a plate, it will recognize it. So you don't have to manually save, manually record, taking the risk to forget to do something and losing your data. Everything will be automated from this point of view. There is a GXP module also if you want to have a better control of your data, more safety, more control, more following. It also measures a lot of different parameters. I will maybe explain you a bit more during all the presentation through all the application I will talk about. But basically, you will be able to follow impedance, capacitance, etc., etc. As I say, Axion do not only provide a hardware machine, but also softwares with different modules. So all these software are very user-friendly in a couple of clicks and in a couple of hours. And actually, I think it was a very nice proof for me during these last 2 days. I show you very quickly how to use the system, and you were totally independent with it. Very nice feature also that we used during these trainings and all the experiments we've been running is the remote app. You can have access remotely to the data from your home, from your phone. So I think it's a very convenient feature. So what is impedance? Since the beginning, I'm talking about impedance. So what is impedance? So in simple terms, the impedance is the resistance that an electrical signal faces as it passes through a material. So in biology, it means that we can use microelectrodes, which are placed under the cells, which are embedded at the bottom of the wells, to monitor the changes in the resistance due to different biological phenomena. So basically here, so I have to figure out how it works. Yeah. So let's suppose this is the bottom of one of the well located in your plate. You can see here there is a certain number of electrodes. When there is nothing on the top of the electrode, the current that we are sending easily go through. So the impedance or the resistance is very low. However, as soon as you will plate your cells, and as long as your cells will keep growing, the resistance will increase. So this is exactly what we are monitoring right here. If at some point, for any reason, it can be natural cell death, it can be a treatment, it can be a compound, it can be an infection, viral infection, the cells are dying, then that will lead to a decrease of the resistance. And this is all of these that we are monitoring over time to assess what's going on in your plate from a biological perspective. For these, you need specific plates, and this is what I'm representing right here. So these plates are, as I said, embedded with electrodes, and there is a very nice feature. As you can see, in the center, there is a small window allowing you to take the plate and to control the cell culture quality at any time point under a microscope. So it can be a classic bench microscope, but it can be also another kind of system, and the one I have in mind is what we call the Omni system, which is a live imager. So later on, I will show you how we can combine impedance assay with live imager assays and what information they can bring all together. So I think for me, the most interesting part for this kind of impedance system is really the fact that you're going to monitor everything from the beginning of your experiment, from the moment you plate your cells to the end of the experiment. Usually when you want to make a cytotoxicity assay, when you want to count your cells, you have to define, sometimes from an arbitrary perspective, an endpoint. It can be 24 hours post-seeding, 48 hours. Why? Because you saw it in a paper, because your laboratory always check at 48 hours post-treatment, and you might lose a lot of information. So to prevent these, I think it's very important to be able to keep tracking about what's going on in your wells from the beginning to the end of the assays. And that's exactly the philosophy of the instrument that you can see here. In this simple experiment, you can see that we've been plated cells. We let them grow for a certain amount of time, 24 hours, and then we had different compounds. It doesn't matter the name of the compound here, it's just for illustrating what I'm talking about. But you can see that all the dynamics is different. If I did choose a time point after 24 hours post-treatment, you can see that you have different effects. But if you check at these two conditions, for example, at 80 hours, you will say, okay, there is not that much difference. So you will lose a lot of information. So that's very important to be able to keep track of everything from the beginning to the end of the experiment. What is very important also is this is entirely label-free. You don't need to add any dye. You don't need to stain your cells. You don't need to optimize the concentration of the dyes or the compounds you want to use for the readout. Everything is label-free. Seed your cells, put the plate inside the instrument, run away. That's how it works. As it's label-free, obviously it's not impacting the biology. You know, sometimes I have in mind people who want to make cytotoxicity assays, they use caspase dye or annexin 5 dye or propidum iodide dye. All these dye can impact itself the biology of the cells. So having a label-free instrument is a very plus. And as I say, no optimization coming from the label of the dye. Since the beginning, I'm talking about the hardware, the system itself. Keep in mind that for every system, there is also what we call modules. So the software which come along with the system. So depending on the experiment you want to do, you might want to choose several modules. So for the impedance, it's very simple. There is just 3 different systems. When I will talk about the Omni system, so the live imager, you will see there is way more depending on, do you want to work on spheroids? Do you want to make conferency? Do you want to make cytotoxicity, etc., etc. So just keep in mind that the instrument is not the only thing you have to think about, but the module which will enhance your science through different applications is also something you have to think about. The plus, I think we already talked about that. You're going to look at the kinetics, so you will not miss any data. This is entirely automated system. Put your plate, run away, have your informations. Level 3, you don't have to stain. GXP compatibility and the app for controlling. Even if this is something I do not recommend, you can check from home how your experiment is going. So now I will try to show you through a lot of different applications all the capacity of the instrument, what kind of information you can extract, what kind of assay you can do, and maybe that will ring a bell in your head and you will say, oh, that's maybe something I can do in my lab. So don't hesitate to let me know at the end of a talk if you want to talk and to go a bit deeper into these assays. So the first one I will go through is the immuno-oncology. So the last decade, it has been the main event regarding cancer treatment. It has been a very novel way to approach diseases, especially cancer, through the usage of individual cells to modify them to enhance the targeting to cancer cells. So I will describe the kind of assay you can do. The workflow is very simple. And you will see that in most of the assays I will describe, the workflow is very simple. You plate your cells. In that case, it was U87 cells. You add an effector cells, whatever it is. It can be a T cells. It can be NK cells. It can be PBMCs. It can be a CAR T cells. And you wait. And you look if the cells are killing your cancer cells or not. In this example, very simple example, you can see that we've been plating the U87 cells. We waited something like 30 hours, and then we added the cells or not. And you can see that when you look at the curve where the T cell has been added, you have a decrease of the impedance. If you remember, if I have a decrease of impedance, I have less and less cells. So a cell death is induced. Okay, so my T cells are killing my cancer cell specifically. If I want to increase a bit the complexity of the assay, we can start playing with different ratio of T cells and target cells. And this is exactly what we have done here. If you look at this curve over here, so we are looking at the cytolysis, we are not looking at the impedance anymore. And that is something actually I can precise in the software. We can represent the data from a lot of different perspectives. You can look at the impedance, but you can also make automatically calculation regarding the cytolysis. So you're looking at what is the rate, what is the percentage of cells which are dying after a treatment. So if you look at here, you can see that we use different ratio between the effector cells and the target cells. And the highest the ratio is, the faster the killing is, which makes sense, right? The more T cell you have, the faster the killing is. Another kind of assay, more and more famous, I would say more and more used, is what we call the CAR T. So CAR T are specific T cells which have been transduced for them to express specific receptors. So in these assays, we use the SCOF3 cell line as a target cells, and we added different CAR T cells. So CAR T cells which has been transduced with the HER2 receptors, CAR T cell which has been transduced with the MOC receptors, so not targeting anything, and T cells which has not been transduced at all. If you look at the untreated condition, which is the orange line over here, you can see that there is obviously no effect. The cells keep proliferating. as soon as you had the T cells, which has not been transduced, you can see that there is a slight killing occurring, or at least the cells are not proliferating. So these flat curve can be explained by there is as much killing as cells which are proliferating. Does it make sense? Yeah, perfect. When I look at the curve where I added the CAR T cell transduced by the MOC receptor, it's exactly the same thing. However, as soon as I use the CAR T cell, which is specifically expressing the receptors of the SCOV3 over here, you have a very drastic killing, very fast killing. So all this automatic calculation allow you to make the distinction and to look at specific killing in that case. In the software, you can not only look at the entire dynamic, but you can also define and look at the data at different endpoints and choose. Is it me? Oh, okay. So this endpoint can have a lot of different usage, like the cytolysis calculation. I will show you during the data analysis that you can also calculate the IC50. If at some point you use different treatment, different compounds at different concentration, the software is able to calculate automatically what we call the IC50 or EC50, depending on the metrics that you look. So far I've been talking about 2D cell culture. Okay, but keep in mind, you can even increase the complexity of the assays. So in this slide, I'm showing you that you can not only do T-cell killing assay on monolayers, but you can also work with 3D objects like spheroids, organoids. And if you look at the dynamic of the killing, which is represented over here, I think I have actually another slide. Yeah, perfect. If you look at the dynamic of a killing, so I will not enter into the detail of the assay, just I give you the global information. Here, this is exactly the same idea. We took organoid and we had a different ratio of T cells, and we look at the speed of the killing. If you look and if you compare the speed of which the T cells are killing the spheroid, you will notice it's way slower, and that's normal. On a 2D, all the T cells are directly access to the cells. When you are talking about the 3D objects, there is a notion of migration, invasion. So this is why the killing is slower. But this instrument is perfectly capable of monitoring any killing also on 3D objects. And here, it can be for cytotoxicity. You can also monitor proliferation of 3D objects, et cetera, et cetera. So sky is the limit, as I say. Very briefly, you can also, this is, let me think. Oh yeah, we talk about 2Ds, adherent 2D. We talk about 3D, spheroid, organoid. We can also make non-adherent cell assays. So it will require a couple of extra steps because the idea is to tether these cells at the bottom of the plate, but it also works. The second most used of the instrument is cell proliferation. And I think in most of the time, that will be the first kind of assay you will run with the instrument. In our case, what we did is we just plated cells and we let them proliferate in and look at the profile of proliferation. So most of the time when people want to characterize their cells, this is the first thing they do. They take a plate, they seed some cells at different density, and they look how fast they're proliferating. What is the proliferation rate? When do they reach confluency? If there is some cell-to-cell contact, which is built over time between the different cells, et cetera, et cetera. So all these things can be assessed with the instrument. So the workflow is more than simple. You plate your cells, you put the plate inside the instrument, you let it go, and this is the kind of profile you can have. So here as information, because I have to be honest, nothing is written on this slide, the different colors are indicating different cell density which has been plated. The darker the color is, the higher the cells amount plated was. Okay, so you can clearly see the different dynamics. These ones are barely proliferating, and these two over here are already reaching a plateau. So it means that they reach confluency. And here, depending on the cell type you're working with, many things can happen. Sometimes the cells are very happy to be held together, and they will keep growing, proliferating, they will start building multi-layers. Everything's good. But sometimes they don't like to be very dense, very dense. So it will start having what we call cell-to-cell contact inhibition, and cell will start dying. And I think this is exactly what we start seeing right here. If you look at the highest curve density, you can see that the curve is slightly decreasing. So it indicates that the cells are starting to die. So if you want to characterize your cell line, it's more than simple with this impedance instrument. Each cell type, sometimes one of the questions that scientists are asking me is, what is the correct impedance value for my cell type? And there is no good answer because each cell type is different. Each cell has its own size. Some are small, some are big, some are a lot of lipids in the plasma membrane. And all these parameters can change the impedance value. And this is what I'm trying to illustrate on this graph over here. So I just took a plate, I plated 3 different cell types, and as you see, the impedance, maximum impedance value are very different between the cell types. So that's also why it's interesting for you to make the cell characterization from an impedance perspective. And also the dynamic is very different. You can see that these ones are barely proliferating, or the division rate is very, very slow. However, when you look at the Calu3, they are proliferating like crazy. So that's also nice for you to be aware of that, because it will save you a lot of time in your assay. If you just need to wait 12 hours for your cells to divide, you don't have to wait 1 week for making your assay. So it will save you a lot of time knowing all these information. Third kind of assay, I think that's the third. Yes, that's the third, is cytotoxicity. And this is exactly what we did here at the NMU. The workflow, once again, is very simple. You plate your cells, you wait for them to reach concurrency, and then you add your compounds. You add your compounds, you wait as long as you need, and you follow the proliferation and the viability of the cells. So in this experiment, we took some A549 cells that we plated for 24 hours. So you can see we reach this very nice exponential growth phase. And then we decided to add a couple of compounds. So we added the Bretfeldin A, cisplatin, doxorubicin, and methoxsantron. So if my memory is correct, so this one is ER Golgi apparatus inhibitors. The Bretfeldin A inhibits the transport of a protein from the ER to the Golgi. The cisplatin, this is a DNA intercalator, so it will prevent replication and it will damage the DNA, leading to cell death. Doxorubicin and methoxsantron, here once again, if my memory is correct, they are also DNA intercalator, and they are inhibiting the topoisomerase 2, if my memory is correct. So once again, DNA damage, cell death. What is interesting to see in this kind of assay is the dynamic of killing of all these different compounds. So depending on what biological part of the cells you're having an action, you see the dynamic is very different. So the methoxsantron killing very, very rapidly the cells. The Bretfeldin A bit more slowly, etc., etc. So you can make a very deep characterization of all your compounds and also all your cells very easily with the impedance system. One key metrics when it comes to cytotoxicity assay is what we call the KT50. The KT50 is the time that your compounds need for killing 50% of the cells. So usually it's a bit like the IC50. This is a key value to know for your compounds and your cells. And the killing time 50 is also a very key value for you to know in this kind of assays, which make me think that we did not calculate any KT50 in our case. That might be something we can keep in mind. Scratch assays, very nice one. I love scratch assays. So scratch assays is the assay in which we try to assess the migration potency of a cells. Okay, so the idea is to plate cells reaching concurrency to make a wound. So you can take a tip, you make a wound, or you can also use a wound maker. And so you will have a gap in your cell layer, and you will look over time the ability of a cell to migrate in this wound. So this is basically how it works, and this is basically what I'm trying to show here. In that case, we used two different, actually one cell line. I think I have a better slide just after. Yes, I think this assay is better. So in this assay, we use two different cell lines. So the MCF7, which is a breast cancer cell line, very low invasiveness. And another one, the HCC 1806, which are a very aggressive cell line in metastatic phase of cancer. Oh, my cursor changed. Okay, perfect. So as you can see here, so we normalize the data. So you can see that everything is at zero, which means here we have 100% concurrency. And at some point, we made a wound. So this is why you have this huge decrease in impedance. So we wound the plate, and we look over time the ability of the cells to recover, to migrate in the wound. So when you look at the MCF7, you can see that there is no changes over time. So the cells do not migrate. And on the contrary, when you look at the very strong invasive cancer cells over here, you can see that very short after the wound making, the cells are migrating, and we almost reach 100% concurrency as we had before the wound. Okay, so you can make the distinction between the different invasiveness of cells in this kind of wound making assays. So there is a lot of things to keep in mind in this kind of assay, but that could be a part of the discussion later on. Like you want to make sure that what you're looking at is really migration and not proliferation of cells. So there is a lot of things in the protocol we can discuss. What is also very nice is to confirm. I always like to confirm what I see on graph by my eyes. So every time I like to take my plate and to go under a microscope. And if you can use a live cell imager, it's even better, because instead of having an endpoint, as you can see here, you can hold the whole dynamic, and you can look your cells actually from the border of the wound going inside the wells, migrating and invading the wounds that you made. So to confirm that here the MCF7 are not migrating at all, we took some pictures just from the moment of the scratch and 48 hours later. And as you can see, these pictures do confirm that there is no migration observed. However, for the HCC, when you look at the pictures, so just after the scratch, nothing in the scratch, and 48 hours, you can see that the width of the scratch decreased a lot. So the cells just migrate. Another kind of assay, what we call cell signaling, more complex, more sensitive, but this is also where the impedance system can play an action. Something that I didn't tell you is the impedance system takes a recording every minute. Every minute, we are measuring what's going on in all the 96 wells. Okay, so that's a lot of information. So when you're using a very slow acting drug, not really important, you will not miss any data. But when you want to make cell signaling, it's very important to have recording very, very often, because cell signaling occurs very fast in time. So cell signaling, I think the most well-known is what we call the GPCR assay, or the RTK signaling. Consists in modification of pathway leading to the change in conformation of the cells, behavior of the cells, cell-to-cell contact due to transduction of signals. And usually for GPCR, it goes through what we call the G-coupled protein. So it occurs very, very rapidly in time, and this is what I'm trying to illustrate right here. I think I have, once again, a better experiment. So in these assays, we use a certain cell type that I didn't write on my slide, sorry about that. And we use different compounds. So once again, we normalize the data here. So we have 100% concurrence, and we treat it with different compounds. So here, once again, if my memory is correct, the histamine compounds is playing a role on the H1 and H2 receptors of the cells, which leads to conformational change. So the cells, usually when you treat them with histamine, they become rounded. The cytochalasin D is an actin inhibitor. If I'm telling something wrong, don't hesitate to let me know. I think that's an thank you for confirming. Actin inhibitors So the morphology of the cells will change drastically. And here, to be honest, the isoproteinol, I don't remember, so let's ignore that one. But as you can see here, depending on the different compounds that you add on the cells, the dynamic and the morphological change over time are very different the one with the others. When you look at the histamine, you can see that there is a very fast effect on the cell morphology, but this effect is very transient. You almost in couple of minutes recover the initial state of the cells. However, in some case, like this is the case for the cytokalasin D, it's a very stable and a very strong compound. So when the cells are treated with such a compound, they change morphology, but it's very stable over time. So this is why the cells never come back to the initial state. Okay, so you can really characterize these cells like these. And if you have a microscope next to you, you can even go further. So you can already imagine how the synergy can be between different technologies. Virology, very interesting one, especially the last years with all these COVID, SARS story. This instrument has been extensively used for making COVID characterization, but not only. Most of the time, people are working for making virus titration because you're able to keep track about the cell viability. You can look at the viral titration. Antiviral screening, you can easily assess if a drug is protecting from an infection. Vaccine development, looking at antibody neutralizing assays, also with antibody, are my antibody able to prevent viral spread, viral infection, cell death, etc., etc. Virucidal cell efficiency, are my compounds able to directly kill my virus, and also oncolytic virus development. So I have very quickly a small example. Once again, the workflow, very simple. You plate your cells, you add your virus, and you look at what's going on. In the first assay I wanted to talk about, it was just after the COVID pandemic. Some team here from the bachelor team made some characterization on how the COVID can spread and leads to cell death. So in these assays, they used, what did they use? Cell type, viro cells. Viro cells, and they infected these cells at different MOI. So all these colors over here are just different MOI. And so you don't need to look into the details. Actually, here you have the endpoint summary, but the idea is the more virus you use, the more cell death I have. Okay, so far, so good. Later on, they made a deeper characterization. They wanted to assess the effect of an antiviral, the remdesivir, to see if it was protecting from the COVID infection. So to do so, they took, once again, viro cells that they infected, and they added different concentration of the antiviral over here. And as you can see, the more, once again here, mini curve, I'm going to summarize it, but the idea is the more antiviral you use, the less cell death you have, viral-induced cell death you have. Okay, so there is this protection effect of this remdesivir over here. As I told you before, one of the nice features of the software, coupled with the Axion BioSystems impedance assay, is this ability to calculate automatically the IC50 for your different compounds. I think that's the last one of the assay I want to talk. I hope in some of these you recognize yourself and you say, okay, maybe that could be interesting to dig into. So the TEER assay, so TEER, it does stand for transendothelial or epithelial electrical resistance. So the main idea of this kind of assay is to not have a look at if my cells are living or not. The idea is to look at, are my cells talking to each other? Are my cells contacting each other? How strong these contacts are? And to do so, we are using a very nice feature of the instrument. Until now, I told you that to monitor impedance, we are sending a current through these electrodes. But actually, we are sending three kinds of currents, three frequencies of currents. One very strong, and the very strong current will go directly through the cells. Remember my first slide when I was showing you the electrode with the cells on the top? My current is going directly through the cells. And so the information that the software will tell me is, do I have something or not? But there is a second current we are sending, and this time very low frequency. And this very low frequency current, instead of going directly through the cell, is going all around the cells. So it will give me information about how my cells are contacting each other, how strong these contacts are. And this is all the main idea of these tier assays. So as an example, I show you two different cell lines here. So what I call resistance is the high frequency. Do I have something or not? And what I call the tier here is the low frequency. Remember, contact junctions. If you look at the two cell lines we tested here, the CALU3 and the A549, you can see that they are nicely proliferating, different rate, but after 20 hours, we reach confluence. So far, so good. However, when you look at the other frequency, you can see that one of them is building contact junctions and the other one is not. Long story short, not every cell type is making contacts junction, okay? I don't know if you remember, in your brain, you have what we call the blood-brain barrier, very important for preventing infection to go from the blood to the brain. So these cells, these epithelial cells, are making very strong cell-to-cell contacts. Some other cells, neurons, blood cells, T cells, they are barely touching each other. They never talk to each other. They never make contacts. So this is a very nice way to first characterize your cells. Are my cells are making tight junction, yes or not? And you can even go further. If yes, you can start playing with different compounds, like, for example, the cytokalasin D, actin inhibitors, as we say, but also the ethanol. So the ethanol is very specific compounds, which are compounds, if I may say, liquid, call it however you want, playing a specific role, especially on the Claudin one, which is a protein which is involved in the tight junctions. And you can see that we plated the cells, we waited for reaching confluency, we were waiting for having these very nice cell-to-cell contacts, and then we treated the cells. And what's going on? When you use cytokalasin D, because you're disrupting the actin, you are disrupting entirely the cell-to-cell contacts. Okay, so this is why you have this huge drop in the impedance over here. And the cytokalasin and the ethanol, on the contrary, is playing a slower action on the cell-to-cell contact, but it's still decreasing the ability of the cells to talking to each other. Brief summary about everything I said. I hope I convinced you that the Maestro is very convenient for putting a plate and running array, and it will do everything instead of you. You will monitor a lot of things. Every minute you will have a data, a time point. You can leave a plate for days, for weeks, to be honest, even for months. People who are working with organoids, organoids, you need time to create them. So people can leave the plate for months inside. The only thing that you have to keep in mind is changing the media from time to time, adding water from time to time, stuff like that. So you will gather a lot of information and all the calculation. Once again, I think it's very important because sometimes you're working with instrument, providing a lot of data, but you have to analyze afterwards. And you will spend hours to analyzing. And that's a pain. Here, everything is automatic. Hands-free collection data, this is exactly what I say. Label-free, I told you, you don't add anything. It just cells and nothing else. And the app, if you are bored one day at home, there is nothing on TV, you can take your phone and look remotely at the data from where you are. The second kind of instrument I want to talk about is the live imager system. So I just have a couple of slides, no worries. I think it's a very nice instrument family, very complementary from the impedance assays. So I can describe them as three different instruments, what we call the OmniBR for bright field, OmniFL for fluorescence, and if you need even more throughput, we have what we call the OmniPro 12, which allow you to run up to 12 plates in the same incubator. Couple of specificities. It's pretty small instrument as well. I would say it takes one shelf on your incubator, so pretty small as well, pretty light. So you don't have to dedicate an entire incubator for it. You can, some people even place two of them in the same incubator. So no problem at all. It will not perturbate the airflow, no condensation generated, stuff like that. So very, very convenient instrument. How does it work? You have here on the center this glass where you will put your plate. Actually, I'm saying plate is not correct. You can do flask, plates, microplates, slides, petri dishes. Everything you have in the lab can be working on this instrument. So you plate whatever you want in the center, and this camera will move around and take a lot of pictures. So as you can see here, we were able to monitor the entire plate by taking roughly 8,000 pictures in 10 minutes. So a lot of data, entire well imaging, different mode of reading, bright field, fluorescence. Talking about fluorescence, you have two different channels, red and green, roughly corresponding to the GFP and RFP wavelength, roughly I said. You can choose to take the entire wells. You can choose to take what we call pattern scan if you want just small part of the wells. And once again, it's a cloud-based. Sorry, I forgot to say. I think it's very nice. This feature is very nice. So which means that every time a picture will be taken, it will be sent on the cloud. And I think it's very convenient, especially for live cell imager, because most of the time when you're using a live cell imager, you're taking a lot of pictures. Remember, 8,000 pictures. You don't want 8,000 picture times, I don't know how much on your computer. That will require a lot of storage. So here, the fact that we send the picture on the cloud will save you a lot of space on your server or computer. Second advantage of sending the picture on the cloud is, so this is an AI-based software. So we train the software regarding different applications, concurrency, spheres, cytotoxicity, whatever you have in mind. We have different modules for each of them, and they are AI-based, AI trained, sorry. So we send a lot of pictures, we train them, so the software know how to recognize the cells, a living cell, a dying cell, a spheroid, etc., etc. So we're sending all these pictures on the cloud, and all servers are analyzing the data. So once again, if you do that, if I do that on my laptop will just die. That's way too much for him, and it will take way too much time. So here it will save you space and time. What can I tell about? Yeah, the Pro 12, very nice. So basically, how does it work? You have this shelter over here, and you have the Omni at the bottom. You have a robotic arm which will take each plate and place it on the center. This plate will be recorded, and at the end of a recording, the arm will place back the plate at the initial emplacement, and it will go to the next one, next one, next one. So all these things can be scheduled through the software which is provided with this instrument. You can tell, okay, please record my plate every hour, every day, every 7 days. Look at the bright field. This time, look at bright field plus red. Whatever you have in mind, you can customize the recording with you. I was talking about the different modules. So this is what I'm trying to emphasize right here. I was talking about the concurrency module, the scratch assay module. Sometimes I think personally, I even prefer making scratch assays with microscope. Like you see your cells going through the wounds that you made. Very, very, very nice to see. You can even make movie. When you make movie from all the picture you took, you see your cells migrating, etc. I think that's very nice. So we have a scratch assay module with the Omni system as well. Clonogenic assay, you can keep track of if my cell is coming from, if my colony is coming from a single cell or different cell, etc. Organoids, as I say, you can work on 2D as 3D. Pluripotent stem cell iPSCs, and for fluorescence, that's basically the same thing, concurrency, and you can also quantify having a lot of information regarding the fluorescence of your object, intensity of fluorescence, confluence of the fluorescence, etc., etc. Why I was saying that it's very complementary, because the application you can do with the live imager are basically exactly the same as the one you can do with the impedance system. So cell growth, cell death, immuno-oncology, drug testing, stem cell discovery, etc. So having this in mind, having this in mind, Axion wanted to develop what we call the connected lab. So the idea of what we call the connected lab is to combine information coming from the both instrument I just presented you, both impedance system and both imaging system. And this is what you can see on this small movie. Here you have, so I have to tell you because you never saw it. This is the impedance software you can see over here. And as you can see, you can have access remotely to the pictures that you took with the Omni. So in the same document, you will have all your experimental condition with the impedance, all the curve, all the IC50 calculation, all the cytolysis value, and also all the picture you've been taken with the live imager, which is combined. So very complementary, providing a lot of information accessible from one single document. I have a couple of slides to show you the kind of picture you can get, to show you how the algorithms are working. So this is a picture of an entire well. So once again, we're taking many, many pictures. In blue, so they are fake colors because here I'm, oh, this is a video. You see, I'm discovering things at the same time. If I can find my cursor. So here you can see a very nice proliferation assay. So what you see in blue is the mask. So in order to be able to detect the cells, the software is calculating a mask, and the variation of a mask are representing the ability of a cell to proliferate or to die. In that case, just proliferation. And after 40 hours, you can see that we almost reach concurrency. I think this feature is very, very nice. I remember when I was in the lab, one of my biggest concern was every time I was making an experiment, you know, when you start an experiment in a flask having 20% concurrency, and when you start an experiment with a flask having 85% concurrency, the behavior of the cell will not be the same. So they will take more time to start proliferating, etc. And I noticed, way too late, unfortunately, that if you normalize the concurrency of the flask you're working, you will have a way more homogeneity of your data over time between all these your experiments. So I think the first application I have in mind regarding this feature is I take my flask, I put it under the Omni or Omni Pro 12 or whatever Omni you want to work with, and you look at the concurrency. So you can normalize, say, okay, from now, every time I will do an experiment, I will work at 40% concurrency. That could be an example of application. I do have a couple of pictures, maybe even a movie of the scratch wound. So as I say, it's very nice. You can create movie with the Omni. So here at the beginning, will it play again? Oh, it's very short. So sad. At the beginning, you can see in blue, you have the wound that we created with a tip, probably over here. And you can see that over time, the cells are migrating into the wound. And so you can quantify the speed at which the wound is filled. We did a small experiment over here. We took these cells and we treated them with PX, which is a paclitaxel. My God, paclitaxel. You see what I mean? Paclitaxel. You see what? Thank you. I think one letter was missing in my words. So which is an so this is, long story short, this is microtubule inhibitors. So what's happening when you treat your cell with microtubule inhibitors? Obviously, they're not able to migrate anymore. So this is exactly what we are looking at right here. Here you have the untreated condition just after the wound making. As they are not treated, the cell just migrates. So this is why we don't see anything anymore. And the more PX we add, the less the cells are able to migrate, just because we are inhibiting the microtubule formation. Okay, so that could be a very nice example of assay you can do with the system. Finally, I wanted to present you a couple of fluorescence pictures. I think it's very, very nice, especially for microscopy, to have a fluorescence. you can do whatever you want. Most of the time, people are using dyes for making cytotoxic experiment. So you don't need it, as we saw with the impedance. You can do it entirely label-free. But sometimes when you want to go deeper into the characterization, it's nice to add dyes. For example, have you heard about the caspase 37? Annexin 5. So all these dyes are, very specific from some steps of the apoptosis pathway. Very early in the stage of the apoptosis, you have these, if my word is correct, the flip-flop of the phosphatidylserine at the plasma membrane, which can be stained by the annexin 5. So by adding annexin 5 into your plate, you can make the distinction between the early stage of the apoptosis or the late stage of apoptosis. Is it even apoptosis? If you have no staining, maybe this is necrosis or whatever cell death pathway is involved. You can use propidum iodide. So this is just a DNA intercalate, so it will stain basically everything which is dying. But you can see, you can make distinction between the different pathway, cell death pathway, which are involved in your assay according to the different treatment, drugs, infections that you want to do in your experiment. And that one, I will not talk about it, just to tell you that you can also easily take pictures, and I'm talking about it, sorry about that. You can easily take pictures about 3D objects, as we did on the impedance system. You can measure 2D, 3D, no problem at all. I actually have no idea of what time is it and how long I talk, if I talk too much or not enough. So anyway, thanks a lot for your attention. It has been a pleasure once again to have you here. If there is any question, please let me know. Preston Krivani will be also here for any question regarding everything that I've been describing over here, especially plates, products, etc., etc. We have the instrument here at the NNU. So this is South Campus, right? So I guess the other one is North Campus. Okay, damn. So the instrument is on the other campus. If you have also any question regarding the assay we've been running with Professor Roux, I think don't hesitate to talk to her. She will be very happy to give her her feedback, feeling, etc., etc.
Are there any questions that you want to ask now?
I just wanted to ask, since the system measures against time, right? And let's say maybe cells and applicated also one of the slides that there would be a cell that which then would observe that the resistance thing. So I just wanted to know if does the impedance detect viable cells or dead cells.
I'm sorry for interrupting. Do we have a mic? I'm getting old and my ears are not working anymore. We don't have a mic. So
she wants to know whether you can actually see on the impedance how many dead cells there are versus.
Okay. So your question is, can you quantify the living cells and the dead cells on impedance?
Yes, because you said that it measures resistance rate by line.
Yes.
And then if the resistance decreases, let's say you maybe treated your cells, and then that will indicate cell death. So I just wanted to know, does then the impedance system detect viable cells or dead cells?
Okay, so what you're going to monitor all the time is living cells, because what the system quantifies is everything which is touching the electrode. And what is touching the electrode is living cells. At least in some extent, before the cells entirely die, it will get rounded and then it will detach. But the dynamic is this one. So you will be always looking at the living cells. But by making a comparison between a control and a dye or a compound, a treatment or infection, you will be able to make a relative comparison between your two assays. So you will say in one case, I have 100% viability. On the other one, I have 80% viability. So my cell death is 20%. Now, if you want to know exactly how many cells died, the impedance is not providing this information. And this is also why no, it does not. It will give you resistant information only. If you want to, there is some tricks we can use to have an idea about how many cells are dying. The first thing that I have in mind is to build a standard curve. Okay, so you take a plate, you seed your cells at different cell density, and you look at the corresponding impedance value. So you know that when you have 50 ohms, it means that you have, I don't know, 10,000 cells. So if you see a delta of 50 ohms, there is high chance that there is 10,000 cells dying. So that could be a first indication. It's not absolutely correct what I'm telling you right here, but it would give you an idea about how many cells are living, how many cells are dying. I think the best approach for answering your question is to combine with imaging in that case.
Often find that the edge cells sometimes they have different data from the material. Is there a way that this platform can actually avoid that, or is just platform?
So that's a good question. That's something you can often see on different instruments. And here I hope you will be confirming we did not have any edge effects. So remember I told you this instrument is, you can control the environment. But if you remember, I talked about the temperature, I talked about the CO2, but there is a third parameter that an incubator is regulating that I didn't mention, which is humidity. Yes. So to prevent edge effect, what we usually like to do is in the plate, we add some water on the edges and it will generate this humidity environment. So here we did not have any edge effect. So as long as you keep the rules, temperature, CO2, and humidity, you will be good. You will be good.
Okay, thank you for the presentation and the training during the time. Quickly, you mentioned now the different tests you can do, like the cytotoxicity and the cell signaling. Can you do both of those experiments on the same plate and in the same well, or because it's different currents that's being sent through the cells?
So when you talk about cell signaling, we are talking about GPCR here, right? Not the tier. Okay. No, that's a question. We are talking about GPCR, and so all the signal transduction from a plasma membrane receptor inducing changing in morphological.
Yes.
Yes.
Yeah.
Okay, that's what you're talking about. Okay. So for doing this, we are looking at the high frequency. Remember, I talked about the low frequency, high frequency.
Yes.
So we're looking at the high frequency. For monitoring cell death and cell viability, we're also looking at the cell frequency, at the same frequency. So here you have to make your choice. In the same well, you cannot make the distinction. So you have to choose, or I look viability, or I will look at the GPCR. However, because you have here a 96 well plate, you can just divide the plate and look at the two different parameters in different.
Perfect, thanks.
Yeah.
I think in that case, thank you very much.
Thanks to you.
Really thank you for all the time that you've been here with us.
That's been a pleasure. That's been a pleasure, and hope it will really keep this collaboration going forward.
Yes, yes. And if there are any questions, then you can either contact me or you can see on our website. There you will find my contact number and also about it.
Did you want me, we talked about looking at the data we've been running together. Would you want me to go through it?
If there's anybody, we actually over the past 2 days, we ran an experiment where we first have cell density, growth curve, and then treatments. So it is, yeah, we can quickly.
Yeah, absolutely.
If you are in a hurry, you can go.
So this morning, so as we said, last couple of days, we ran 2 experiments. And this morning, you sent me the data. I had a very quick overview of them. So if you're okay, I want to show you how the software works and how to run analysis to show you how easy, I hope that would be easy for me to make the analysis.
This case it was expected. So in this case, this was a. Okay. Yeah. So in this case, this was a real experiment that we've done over the last 2 days. And this was just, he will tell you what we have done. But yeah, we did not know what our results will be. And like for incidents, I think it would be nice if one seeded the density of the cells according to the standard curve that you do beforehand. But because we only had 2 days, we actually did it on the same plate. And then you can see how, whether we seeded it at the right density.
Do I need this one? No, I don't. So indeed, when we have time, when I'm not in a rush or just sitting here 2 days, the correct workflow for this kind of assay, which was a cytotoxicity assay, is first what we call running a density sweep. The main idea is because each cell type is different, because each lab cultures these cells on its own specific manner, the dynamic of the cell growth can be different. So what I like to do the first time I come on site is to run a density sweep. We take a plate, we seed the cells at different cell density, we wait 24 hours, and we look what's going on. So that will already take 24 hours. And then the second part of the experiment is the drug testing. Because we were running out of time, we decided to combine the 2 assays in the same plate. And we were hoping for having the correct cell density straight away. We were very close. So the first part of the assay was the density sweep, and this is what I want to display right here. So I don't know if you can clearly see the colors. So this is what the software looks like. You see, it's very simple. You just have a couple of different tabs. You can easily create a plate map. Remember, I told you everything can be automatically calculated. Average, standard deviation, SEM, IC50, cleaning time 50, etc. Everything can be automatically done as long as the software knows what is looking at. Sorry. So we did create a plate map. This is the part that you can see on the top right corner. And I did create a color code. So here what you're looking at is HT29 cells plated at different cell density. The darkest the green is, the highest the cell density is. And you can see we made a gradient, 40,000, 20,000, 10, 5, 2.5K. And we looked at the proliferation pattern of these cells over 24 hours. As you can see, there is a very nice dose response of the impedance according to the cell density. So the highest the impedance is, the higher the cell number we plated was, okay? And when we see that, because in this kind of cytotoxicity assay, you want your cells to be super happy, but not too confluent. So you want to reach this sweet spot after 24 hours. And look at this tree curve, way too low, they are barely proliferating. Not ideal, not ideal. I mean, we took the 10, so. This one, the 20,000, very good. We have this nice exponential growth phase, and this one, very good as well. However, if you look at the end of the experiment, you can see that the curve is decreasing. So, oh, my cells start dying. So I don't want this condition either. So you can see that you can easily characterize the cells. You can clearly see when the cells attach, start spreading, proliferating, etc., and start dying. So in your case, in our case, we took this density, the 10,000, which could have been a bit higher. Like if we have chosen like 15 or 20,000, that will have been even better. But that was okay. But the cells was proliferating. Then for the other part of the plate, what we did is we plated 10,000 cells per well for the HT29, and we added different compounds. So on the top, I'm going to highlight right now. On the top over here, we added different nanoparticles in different conditions: silver nanoparticles, gold nanoparticles, silver particles combined with liposomes, etc., etc. So all these blue colors are different treatments. Okay, and we work as triplicate, so this is why you see 3 wells with the same color for each conditions. And we look at the effect of these compounds on the cell viability. And so here, so this is why this is now that I have to work in real time. So give me a couple of seconds for set it up. So I'm just going to zoom from, is that the good experiment? Yes. So is it me or there is a part missing? No, that's good. So there is.
The one I sent today had around 40 hours.
Oh, I took the wrong experiment. Oh, oops. Okay, so unfortunately, I did download the wrong experiment. So I am going to try to show you stuff anyway. So this one, okay. Why I
duplicate the screen?
Okay. We did export, so to summarize what happened here, we did export twice the experiment, yesterday evening and one this morning. Unfortunately, I have the one of yesterday evening, so I will try to tell you out loud what we had as a result, but it could have been better for me to show you the other one. So what I'm trying to show you right here is what is the effect of the different treatment I mentioned, so the different nanoparticles combined or not with the liposome on the cell viability of the HT29. So you have here the proliferation curve of the cells. You can see these small right arrows, so they are tagged. Here we added the cells, and here we added the treatment. So I'm going to try to zoom in from the moment we added the treatment, and I'm going to try to normalize the data from here. So let's zoom in from this moment we added the cells, which was roughly 20 hours. Okay, and I'm going to normalize the data. Yes, okay, that's pretty good. What I did just right now is, because I had small variation, I say I want to normalize all my data from the moment I had in my drug. I'm comparing to this control, which is the 10,000 cells per well without any treatment. And once again, all these blue colors are my treatment. Here, if I look at my endpoint, I can see that it's a bit early. Once again, we were running a bit out of time. I think we treated the cell yesterday around 3 in the afternoon, 4 in the afternoon. So it's not even, you see, 24 hours treatment. But this is also the beauty of the instrument. You can let the plate go and look what's going on. So now I have access with my phone. I think from France, I will look at your experiment remotely. But you can already see some effects, right? So this is the silver condition. I think if I was taking my calculator and making some statistical calculation, I think there is a statistical differences between the control and this one. For the goal, this one is the goal. I don't think there is any effect, etc., etc. You can see small effect over here. This one has a big effect on cell viability. So already after 20 hours, 20 hours of treatment, we can start seeing effect on cell viability. So that was very, very nice. And on the other part of the plate, so all the wells which are underneath, I'm going to highlight them right now. What we did is we took two compounds. We took the doxorubicin and the 5-FU, which are both DNA intercalants, leading to cell death. And we, instead of using one single concentration, we made a dose response. Okay, so we use 6 different concentrations, and the idea was to try to determine the IC50 of this compound in these cell culture conditions. So let me just change the conditions over here. Perfect. I could even zoom a bit better here. Perfect. So here the idea is it does not make sense, but the lightest, the darker the blue is, the lowest the drug concentration is. Okay, so very high concentration, very low concentration. Okay, and you look at the effect on the cell death. Once again, here when you look at the endpoint, maybe I can even display one time point every minute to make the curve a bit more smooth. All right, way better. You can see that once again, we start seeing something from a dose-dependent manner. So the highest the drug concentration I have, the lowest the viability is. So the highest cell death I have, etc., etc. So in that case, that will be very interesting to keep the experiment going to see if we increase this effect. And every DNA intercalant, most of the time, especially here, we work with pretty low concentration, to be honest. When I look at the literature, that's also a bit my fault. We based our calculation on an IC50, where people looked after 48 or 72 hours post-treatment. So here you see we are very, very, very early post-dosing. So this is why also one of the reasons why we just starting seeing some effect of the drug. But you start seeing something. And one very nice feature of the software I told you is this IC50 calculation. So this is just this tab over here. I want to show you how it works. Let me just remove this. Let me rescale. Here you go. See, that's very, very nice. In just a couple of clicks, you select the wells of your interest, you create a plate map, you look at the dynamic of the viability and the cell death, and you have access to the IC50 curves. So here you have the concentration of drugs. Here you have the, in our case, the resistance, because we're working with impedance. And you have this nice semi-logarithmic curve to determine very easily the IC50 value of your drugs. Very simple. To finish, I want to show you the other drug, but unfortunately, this one was not working as expected, or at least right now. Let me display the control once again. And here you see basically no effect, no effect. So is it because it is too early? Once again, only 20 hours treatment. Is it because I choose once again an IC50? We worked around an IC50 way too low. And after discussion with Professor Wu, apparently they used to work with highest concentration of the drug on the lab. So maybe the cells are a bit more resistant to the compound. Are we too early in the process? Should I have been working with the highest concentration, etc.? So all these questions, we can keep them in mind. And actually some of them can be easily answered by just leaving the plate in the instrument, waiting for a couple of days and looking what's going on in here. So I hope I did convince you that the instrument is actually very nicely designed, pretty easy to use. You can extract automatically a lot of information. Here I did not mention many things, but you see all the average. We have replicate. The software knows what replicates there are. It recognizes them. It calculates the average, standard deviation, SEM, IC50, etc., etc. You can export everything through PDF, presentation, whatever you want. So there is a lot of features. I will stop talking right now, but there is a lot of very nice features for you to enhance your science, to make it easy for you to spend more time wherever you want, but not at the bench.
I think the nice thing that impressed me and the students that worked with it is that they can actually watch their experiment from their phones at home late night. So the software is so nice that you can download it on your computer and that you can do all your calculations at home. You can also follow your experiment from home, etc. And then if something does not go according to plan, and that was on our first plate, we saw something wrong, and then we could immediately stop the experiment right there and start all over, which means in this we have done the same as the MTT and cell density, etc., in 2 days, where it sometimes takes weeks and time to repeat and with this one, we could, the first day we saw, well, it's the first hour.
First hour.
First hour, we saw there's something wrong and we could immediately correct the conditions. So then the experiment for the 2 days, otherwise we saw at the end of the 2 days, we must start over. We could do it immediately. So we are very excited. So on what we are going to do, I suppose all the students would like to go to the lab now.
I hope so. I hope so.
Yes, can we just see that template again? The calculation. How did you normalize that part?
How do I know what?
Normalize the normalization that you did.
Oh, yes. Here in this tab, you can decide a time reference. So I just said I want to normalize over here, and you just select when.
You want to,
Yeah.
just track to the time point you want.
Yeah. And in that case, I choose from the moment we added the drug.
How did you actually want to much whether it took? How do you know the time that it was added?
Oh,
okay, you could put it there, right?
Yeah, because when you on the software, when you do something, you can create some notes.
Yes, I
And so when we added the drug, I wrote a note and I say, oh, here, this is where it happened. Absolutely.
Thank you to everyone
Thank
who's
you very much.
participating online. We are done for today. Is it?
Yes.
Okay. Thank you so much for the online people.